Hi, and welcome to In Search of Insight, Nootropics Depot's monthly podcast. I'm your podcast host, Erica, and sitting next to me is our product specialist at Nootropics Depot, Emil.
Hey everyone.
Today, Emil and I are joined by our science director at Nootropics Depot, Dr. Abraham. Welcome to the podcast.
Hi, happy to be back. And feel free to call me Ellie. The doctor is, we'll keep it more casual.
Awesome.
But the topic of conversation today is about how you did become a doctor and your PhD research specifically into botanicals and machine learning, and how machine learning can help us identify botanicals better in the future. This could have implications for the whole supplement industry and specifically for us because we have a direct source for that research and expertise because Ellie is on our team now. And we can talk about how all of this will come together and potentially make the products that you're taking even better.
Machine learning just as a concept is something I'm not so familiar with, but this is your whole study. So can you give us just a general idea of like what is machine learning and then what did you do specifically within that?
Right. So machine learning is a pretty broad category. It's kind of any situation where you're providing an algorithm or computer data, and then you build the algorithm so it can find patterns within that data. So I'm not going to get too deep into the actual statistical terms because, it'll bore even myself. But essentially, you want to take your big data and see patterns that we would never be able to see. And then you can do things like, based on these patterns from this previous data, what do you think this new data is? So that would be more of a classification problem. So in terms of botanicals and my research, I would say based on all of these different types of basil, these are their chemical profiles. This is all of the compounds in 30 different kinds of basil. Based on that information, I have this new basil that I ordered online. What kind do you think it is? So maybe taking a step back to talk about the basil side of that before we get more into the discussion of the algorithms. Basil sounds like a really straightforward plant. You grow basil in your garden. There's holy basil, that's what we have as teas. But there's actually over 60 different species of basil, which I don't think most people know. And while a lot of those species will have the same, effects when we take it as a supplement, they do have different chemical profiles. And so it was a really good model species because I could grow a bunch of varieties. I grew 30 different varieties of basil. And I could look at their chemistry, but it wasn't hard to grow. So I was able to grow it in a greenhouse, manipulate for environmental conditions, but still get varied chemical profiles. So I used that variation between different varieties and for different species of basil to build predictive models. So those models, as I said, use their chemical profiles to say, okay, this new sample of basil looks the most like sweet basil, or it looks the most like Krishna, holy basil. And so I could say, this basil that is being marketed as a holy basil tincture is actually just sweet basil that you would grow in your garden. Mm. And that's based off of similarities and differences in the chemical profiles. And while for basil, that might not seem so important, you're not going to have adverse effects from taking the wrong kind of basil. When you apply those algorithms to, say, plants like turkesterone, or if we were looking at the difference between two different kinds of ginseng, or plants that are more expensive, that's when those algorithms can have a bigger role. So that we could say, we know based on the chemical profiles what this is or what this is not.
So is this would be a continuation of HBTLC, doing things that HBTLC couldn't do, or is it more like a companion? You would still need to use HBTLC? I read through your study a bit, which, by the way, for those of you interested, we will link it, of course, in the description, because Ellie is really a published researcher as well, because your study got, or your article got, got published in Nature, right?
It was actually in analytical and bioanalytical chemistry.
Okay, nice.
pretty far away from nature,
Yeah.
but pretty big in our specific field.
Yes. Yeah.
So,
Yeah.
and then I have a review for anyone interested in learning more about the actual kinds of algorithms that we use and the different kinds of models. I have a review paper that talks about every kind of model being used for authentication of botanicals, how it's been used, and kind of some future directions. And yeah, we would definitely link that below. And that gets more into the actual details of the statistics. And then, the paper with the basal authentication is a published article as well. And that one, ultimately, we found that doing this kind of machine learning is actually not a sufficient way to authenticate botanicals. So far I've said, we can classify, but it is just an idea right now. This is a new field. A lot of people are trying to use this for authenticating botanicals, but it's not at the point yet where the algorithms are sophisticated enough to do the prediction accurately. And in my specific study, I found that there was a lack of variation when you grew all the plants in the greenhouse. So we weren't encompassing different types of drying, different altitudes of growing the basil, different soil conditions, different pests and pathogens. And all of these different things have an impact on a plant's chemistry. And without encompassing all of that variation, you're never going to have a model that accurately represents products that are on the market. And that's the biggest limitation right now, is having enough authentic material to build models that are actually representative of the marketplace.
Which I think brings up a broad issue that we've touched on a lot on this podcast, is that a lot of what's claimed in the supplement industry isn't actually real. You might be buying Ocimum sanctum, or what's the other Latin name for holy basil?
Tiniflorum, Ocimum tiniflorum.
Yeah.
Ocimum sanctum is actually the same thing. They're just different names for the same thing.
Which one actually is correct, or are they both correct?
I think sanctum is used more.
Okay.
But they're both correct, and they're both the same species. And then there's also Ocimum graecium is the other holy basil species that I studied. And then sweet basil is Bacillium.
Okay. And that's the one for those curious, that's what we eat mostly, right? The sweet basil?
Yeah, if you're making pesto, that's going to be what you're using.
Nice.
What I found really interesting about the study is that, from what I could gather, the method worked for the plants that you grew, but then not for the commercial samples, which brought up a lot of ideas in my head, and things we have talked about in this podcast a lot, is that when we dry, we lose things. When we extract, we lose things. And I thought one really interesting thing in your sample prep is that you immediately put everything in liquid nitrogen, right?
Right.
And Erica used to work at a restaurant in New York called Sandbar, which is being
Momofuku Sanbar.
which is being run by David Chang, and connected to that there was a kind of a molecular cocktail bar being run by a guy called Dave Arnold.
It was called Booker and Dax. It was amazing, but it no longer exists, sadly.
And they had rotovapes to make things, and they had I'm blanking
Liquid
on it.
nitrogen.
They did a lot with liquid nitrogen, and this is one of the things he was
They
doing.
use siphons a lot to create different foams and things.
Yeah, and it was the centrifuge.
Yeah.
Centrifuge they used to clarify a lot of different things. So they would take lime juice, put it in a centrifuge, and then take the fraction out that was completely clear, and then you can carbonate that. You could do all sorts of crazy things with that. But one really interesting thing, and maybe the most interesting cocktail I've ever tasted in my life, was a technique he came up with, which was called cryo-muddling, where he would take fresh basil leaves and he would put them in a shaker tin, and then put liquid nitrogen in the shaker tin, and take a muddler and muddle it all, and then put high-proof spirits in it. And his thinking was that there's an enzyme in basil that degrades the positive volatile aromatics. So if you take a leaf of basil and you crush it up, you break the cell walls, then that enzyme gets released and it degrades the sensory attributes that you would get if you just smell a basil leaf fresh. So he thought at really low temperatures that enzyme can't get activated, and then you break it up and then all of the cell walls are broken, so that enzyme is easily accessible by the alcohol that you then add to it, and the high proof alcohol in the research he did deactivates that enzyme and keeps all of those volatiles there. And it really smelled like the most basil-y thing I've ever smelled. So going back into your study, I thought it was interesting that you're basically doing a very similar process.
Well, it's like almost the exact prep that I would use in the lab. Yeah, we would flash freeze things in liquid nitrogen, just because that really does keep the exact chemical profile of the time you harvest it. And we harvested all of the leaves of the plant for any, botanical scientists listening to this. That's an important part, is we weren't just looking at the basil leaves, but the base leaves. We were looking at all of the leaves, and we harvested right before flowering. So it was very consistent. And so all of the chemical profiles weren't swayed by time. So we wouldn't be seeing differences in harvest time or the type of leaf. And when you flash freeze it, it really contains those compounds. So you're looking at that exact time, what was that chemistry like? Which is great for the greenhouse study, because we were able to use machine learning to differentiate between the different species of basil and the varieties of basil using machine learning. But because we had such high control of those conditions, we weren't able to model anything outside of the greenhouse.
And I think that goes back to with drying we lose things, and then a lot of the things you probably tested were also extracts. I think I read you had some whole leaf stuff, but also some extracts. And in those extracts, you always lose something as well. And a really good way of highlighting this is if you make a cup of coffee, you end up throwing away the coffee grounds. You get an extract and you're throwing away a lot of it, and that's how all extracts work. And that stuff that you're throwing away is probably important markers as well, and bioactives.
Exactly.
So we are more and more now playing around with non-extracted plants, because if you grow them to a very high degree and you can manipulate maybe the cultivars, the growing conditions, you can get very high amounts of bioactives without having to do any sort of extraction and concentration, which then has a higher degree of bioactive diversity. And I think that's also where some of your research comes into play, maybe being able to deal with a higher amount of bioactive.